Hello, my name is Hosseinali Asgharian. I am a postdoc in the Good Lab at UCSF. Today I'm going to talk about Ribolog, an R package we have recently developed to analyze ribosome profiling data to study translation dynamics and regulation. In ribosome profiling, translation efficiency of a transcript is calculated as the ratio of ribosome protected fragments, or RPF reads, to total mRNA reads. Ribolog offers several advantages to the existing methods of analyzing ribosome profiling data, which we will review in the following slides. During translation, ribosomes sometimes slow down on certain positions because they hit a suboptimal codon or an RNA secondary structure or an RNA binding protein, for example. Reads accumulated on these positions do not represent active translation and must be teased out. We introduced the Kelp method for calculating stalling bias coefficients and debiasing the RPF counts. Applying it to real datasets, we have shown that the Kelp bias coefficients correctly capture the dependence of translation rate on tRNA abundance. We have also shown that Kelp corrected RPF counts are much more evenly distributed along the transcript, as demonstrated by the purple bars on the plot to the right. Next, we model translation efficiency as the odds of observing RPF reads versus RNA reads in a dataset. And we model translation efficiency ratio between samples as the odds ratio of a logistic regression with sample attributes as predictors. The output of this regression can be visualized and explored for biological insights. Now to the QC tools. First, we do a principal component analysis of translation efficiency. This allows us to evaluate replicate reproducibility and generate biological hypotheses. For example, on the plot to the left, you can see that PC1 separates two non-metastatic cell lines from two metastatic cell lines, indicating that metastasis is the major driver of translational shift in this dataset. Other QC tools include proportion of non-differentially translated transcripts between samples and correlation of equivalent tests. Meta-analysis is useful when we want to aggregate the results of different experiments, but we cannot directly pool the raw data. For this purpose, we use a modified inverse chi-square method. Applying it to a real dataset, we showed that the meta-analytic framework faithfully captures the translation efficiency ratio represented by beta, or the regression coefficient, and its associated p-value when compared to the ground truth. Next, we offer a tool for empirical significance testing, which is useful to reduce false positive rates when some theoretical assumptions may have been violated in the real experimental setting. For upcoming modules and for letting us know your suggestions and comments, please visit our GitHub page. Thank you.